A COVID booster shot is an additional dose or doses of a vaccine given after the protection provided by the original shots has begun to decrease over time. This video is to clarify how mRNA works in gene transmission as regulation, expression, and transfer of genetic traits, and COVID booster shot has no effect on those processes. The relationship between DNA and RNA is called the central dogma of molecular biology. DNA makes RNA, and RNA makes protein. An organism's genome is the totality of all of its DNA. A cell's genome functions as a computer program. A computer program is copied from ROM, or the read-only memory on the hard drive, to RAM, or the random access memory when you open it. The program is really executed by the instructions in RAM, but the RAM copy of the program only lasts as long as the system is powered on. If power is lost, you must restart the program, which must then be copied from the disk to RAM once more. This setup should protect the master copy of the program from being harmed by a power surge or human error. If DNA is the cell's genetic program's master copy, or the ROM, then it must be kept intact. RNA serves as the working copy, which protects the DNA in one way, which is the RAM. RNA and DNA share a lot of similarities chemically. From a biochemical perspective, the primary distinction is that RNA either functions as a part of the metabolic apparatus or serves as a replica of the instructions for protein synthesis.
Pari ko po ang malaman ng inyong mga misconceptions before nag-booster shot. Opo ma'am, bali po nung bago ako magpabakuna, ang misconception po kasi ay magiging zombie ka daw at syempre isa po sa dahilan ay ikakamatay mo raw ang booster shot.
How well a COVID-19 vaccine shot works varies from person to person. While getting a booster shot will likely give someone more protection than not, a new study finds your genes actually play a major role in whether you're really safer after receiving a booster. Researcher from the University of Michigan says their findings could lead to more personalized vaccination in the future, which boosts the efficiency of the antibodies. What's most interesting with this work is the concept of personalized variability and understanding direct links between vaccines responses and different genes people have, says Kelly Arnold, a UM Assistant Professor of Biomedical Engineering in a university release. According to their findings, antibody concentration may not even matter because that person genes encode for immune receptors which do a poor job of sticking to the antibodies. Scientists call this problem having a lower affinity. As a result, the patient could have a respectable antibody count and still have a weak immune response after vaccination. Although it's just a theory today, researchers says it may be possible to design a vaccine that also tweak the structure of antibodies. This would make them more likely to stick to a patient's immune receptors, overcoming the problem in their genes. Depending on your genetic background, we've found that vaccine boosting may be more or less effective in activating certain innate immune functions, Arnold says. And in some people where boosting the concentration of antibodies was ineffective, being able to change the affinity of antibodies could be more successful route, though this is still a theoretical concept and not yet possible in practice.
Boosters do provide another layer of protection, especially with the rise of the Omicron variant. I think there is broader consensus now among doctors and scientists that everyone 18 and up in the United States who can get a booster should get a booster. That boost won't last forever, but it does buy you time.
Ano naman po ang mga positibong epekto ng booster shot sa inyo? Bali po ang naobserbahan ko ay naging mas malakas pa po ako lalo. Yung katawan ko po ay nag-light kumbaga. Mas malakas po ako pagdating sa maggawa ng gawaing bahay, ganyan. At kalusugan na rin. Thank you.
According to Lisa Maragakis and Dr. Gabor David Kellen of Johns Hopkins Medicine, the protection offered by the previous shots has begun to wane over time, at which point a COVID booster shot is administered. The booster supports in maintaining a high level of defense against serious coronavirus diseases. As of July 22, 2022, there were 68,993,523 administered first doses of COVID vaccine, and there were 71,558,568 for second doses, while there were already 15,945,220 booster doses.
Eli, nakapabooster ka na ba? Ikaw, Evie, nakapabooster ka na ba? Kailan pa?
Matagal na.
Sana all. Ikaw, Joberlin, nakapabooster ka na ba? Hindi pa. Kailan kaya tayo magpapabooster? Ikaw, Tol, nakapabooster ka na?
Di pa nga tol eh. May alam pa ba kayong magpaboosteran?
Sa tingin ko kasi meron nata doon sa sa munisipyo per schedule nga lang yung pagpapabooster. Baka sakali meron tayong schedule na makikita doon. Kaya ano pang hinihintay nyo? Dapat magpabooster na rin kayo. Kaya nga sabi ng DOH, pag nakabooster ka, Pinas lakas!
The appearance of differently colored zones in the leaves and sometimes the stems and fruit of plants. Variegation is caused by mutations that affect chlorophyll production. The striking look of variegated plants is desired by many gardeners, and some have deliberately tried to induce it for aesthetic purposes. There are a number of gardening books about variegated plants, and some gardening societies specialize in them. Because the variegation is due to the presence of two kinds of plant tissue, propagating the plant must be a vegetative method of propagation that preserves both types of tissue in relation to each other. Typically, stem cuttings, bud and stem grafting, and other propagation methods that results in growth from leaf axil buds will preserve variegation. Cuttings will complete variegation may be difficult, if not impossible, to propagate. Root cuttings will not usually preserve variegation since the new stem tissue is derived from a particular tissue type within the root. Some variegation is due to visual effects caused by reflection of light from the leaf surface. This can happen when an air layer is located just under the epidermis, resulting in a white or silvery reflection. It is sometimes called blister variegation. A common cause of variegation is the masking of green pigment by other pigments such as anthocyanins. This often extends to the whole leaf, causing it to be reddish or purplish. On some plants, however, consistent zonal markings occur, such as on some clovers, bromeliads, certain pelargonium, and oxalis species. On others, such as the commonly grown forms of coleus, the variegation can vary wildly within a population. In our case, we variegated Mayana plant or coleus plants with a mother plant of black dragon coleus with red velvet coleus, and this is how we did it. First, we prepared the materials needed for the variegation process. The live and healthy Mayana plant varieties in pots, sharp knife or cutter, and plastic adhesive tapes. Then, we chose the Mayana plant that will going to be the mother plant and other type of Mayana plant that will be inserted in the mother plant. After choosing the branch where we are going to graft, we inserted the cuttings to the branch and secured the stems using the adhesive tapes. After securing the stems, we placed the variegated Mayana plant away from direct sunlight and heavy rainfall and wait for a few weeks. We checked the variegated plant after 2 weeks, and here are what we have observed so far. Variegated plants have long been valued by gardeners as the usually lighter colored variegation can lift what would otherwise be blocks of solid green foliage. These variegated plants have become a must-have for home decor, but masked behind it is the amazing processes happening in their genes. The early peopling of the Philippines based on mtDNA. The past Philippine candidates for Miss Universe pageants proved that there exists a beautiful blend of the Filipino race. The lineages of Miss Universe Catriona Gray, Pia Wurtzbach, Venus Ra, and others are examples of how traits of the original Filipino can compatibly mix with other races and result in such awesome looks. Studying the phylogenetic tree in the picture below shows how the gradual change of appearance took place by tracing from early ancestors to the present-day Homo sapiens. Let's move on with the human evolution, which is illustrated in a cladogram that shows the physical features of our early ancestors such as Australopithecus moving up to the present species of Homo sapiens. In the past, it was incorrectly assumed that human evolution was a relatively straightforward sequence of one species evolving into another. Complicating this evolutionary history even more is the realization that our ancestors very likely mated successfully with members of other closely related species from time to time. As a consequence, our inherited gene pool was enriched by added genetic diversity. The striking similarities in appearance between the human genus Homo and our ancestors, the genus Australopithecus, is sufficient reason to place us both in the same biological tribe of hominini. Both genera are bipedal and habitually upright in posture. Humans have been somewhat more efficient at this mode of locomotion. Like Australopithecines, early humans were light in frame and relatively short. They were only about 3 feet and 4 inches to 4 feet 5 inches tall and weighed around 70 pounds or 32 kilograms. The evolution of larger bodies occurred later in human evolution. The differences between Australopithecines and early humans are most noticeable in the head. Humans developed significantly larger brains and relatively smaller faces with progressively smaller teeth and jaws. In addition, humans became ever more proficient in developing cultural technologies to aid in their survival, while the Australopithecines did not. Early transitional humans had brains that on average were about 35% larger than those of Australopithecus africanus. In fact, it is beginning with Homo habilis that our ancestors finally had brains that were consistently bigger than those of the great apes. People, but not apes, have a gene that stops the production of N-glycolylnuramine acid. Using molecular clock analysis, the UCSD researchers determined that this gene entered the human evolutionary line as a result of a mutation 2.7 million years ago. While it is presumed that the Australopithecines lacked this gene, there is no direct evidence.
What are the migration routes in the early people of the Philippines? So, based on the article entitled The Early Peopling of the Philippines, based on mtDNA, the following are the routes and maps that show the possible routes of early colonization of the Philippine archipelago. First, the northern route, which is from Taiwan through the Luzon Strait, and here is a closer look of its location. Second, southwest route that is from Borneo to Palawan, and this image shows its location in the map. Third, central south route, which is from east of Borneo, crossing the Sulu archipelago towards western Mindanao, and here's a closer look of it. And lastly, southeast route that is from Sulawesi through southern Mindanao through the Sangi Islands, and this image shows its placement on the Earth's map.
Indigenous peoples were present in each of the identified places of migration routes. Migration place in Luzon Strait, Bataan, and Babuyan Islands. The indigenous group of people present are the Aitas and Ivatan. In Bataan, there are small Aita settlements in almost of all the municipalities. In Dinalupihan, Hermosa, Orani, Samal, Abukay, Balanga, Orin, Limay, Mariveles, Bagak, and Morong. The Ivatan group are an ethno-linguistic group native to the Babuyan Islands of the northernmost Philippines. They also share close linguistic and cultural affinities to the Tao people of Orchid Island in Taiwan. Migration place in Palawan. Palawan is the largest province in the Philippines, is home to several indigenous ethno-linguistic groups such as Cagayanen, Tagbanwa, Palawano, Taot Bato, Molbog, and Batak tribes. Migration place in Sulu Archipelago towards western Mindanao. The indigenous group of people present are the Tausug. The archipelago is the home of the indigenous Tausug people. Migration place in Sangihe Islands. The Sangir are the indigenous people, inhabitants of the Sangihe Island chains located in southern Mindanao.
What are the evolutionary scenarios significant in tracing the early peoples of the Philippines? The fossil records and the major roots become significant in tracing the early peoples of the Philippines. The fossil records confirm the existence of inhabitants about 40,000 to 50,000 years ago, and the genus Homo can possibly be 66,700 years ago. The major routes from north and south are greatly affected by last glacial maximum, or the LGM, on which southern routes were more favored by migrants due to low sea level compared to the northern routes. Which evolutionary scenario fits best with the real data of migration to the Philippines? The evolutionary model that depicts migration with long-distance dispersal, or LDD, best fits real data. The Philippines had rapid settlement as a result of rising population. Discovery backs up earlier research on Paleolithic expansion in Eurasia. Under the most appropriate evolutionary model, the population genetic parameters were estimated. The approach demonstrated a performance with overall small estimation errors that always fell within the 50% higher posterior density interval, or the HPDI, with regards to the true value when the parameters estimations were validated. The past proves the beauty of the lineages of the Filipinos mixed with other races. This hidden beauty, like a gem, is hidden in our bloodline through our mtDNA. Discovering ancestors is performed through DNA matching that compares your unique DNA pattern with others in the world's largest mtDNA database. Through the determination of the haplogroups, family tree DNA allows us to trace migration patterns and determine where our maternal ancestors lived going back thousands of years. Today, we are the living example of how these traits of the original Filipinos can possibly be compatible with other races and result in such awesome looks. A herbal soiree, an ointment that is made up of Acapulco leaves, guava leaves, and ginger with paraffin wax and coconut oil. 270 milliliters is the total amount of volume for coconut oil consumed in our ointment, 35 grams for both Acapulco and guava leaves, and a total of 40 milliliters for ginger when it was extracted, and lastly, melted candle or the paraffin wax has a total volume of 300 ml.
According to ICANN in the year 2019, cresofanic acid is a fungicide used to treat skin infections and is present in Acapulco leaves. Guava leaves contain significant amounts of antioxidant, anti-inflammatory, and antibacterial compounds, as well as tannins, which help treat everything from stomach problems to long-term conditions like cancer. Other studies stated the beneficial effects of the guava leaves extract. Based on a study on antioxidant activity of certain plants in Thailand, mechanism of antioxidant action of guava leaf extract. The free radical scavenging and reduction of oxidized intermediates were the mechanism of antioxidant effect of guava leaf extracts. Through reducing processes, the phenolic content of the guava leaf fraction significantly influenced the antioxidant activity. On the other hand, zingiber officinale, often known as ginger, has been used as a traditional medicine in China and India, and the rhizome has curative characteristics connected to digestive difficulties, constipation, and gastrointestinal sickness, according to a study by Citi published in 2020. In addition, ginger has been shown to be a powerful antibacterial. The anti-inflammatory and analgesic properties of curcumin, a terpene, and the spicy aroma and flavor of ginger are a result of the molecule sabinin. One of the components of ginger, known as gingerols, is what prevents the growth of pathogens like Staphylococcus aureus and Listeria monocytogenes. Both Acapulco and guava leaves were collected from Aurora Alicia Isabella. The Acapulco leaves were air-dried for about 1 week, while the guava leaves were dried under the shade within 5 days. The garbling process and air drying immediately followed after collecting the leaves respectively. Using a straw, the Acapulco leaves were tied up and air dried on the grills of our window, while the guava leaves were garbled to remove the extraneous matter and then eventually dried up under the shade. The herbals were guava, Acapulco, and ginger ointment, made and packed by G3.